we're going to give the welcome to our next speaker, plenary speaker, is Jennifer Dionne. I won't delay more this, so I just, everybody knows Jennifer Dionne from Stanford University. For us, it's really an honor to have her here. In fact, she was, since the beginning of SHIFT, in SHIFT 2017, but you remember, you entered Telematic, you remember, online, because it was impossible to travel at that time. But on last edition, she attended the SHIFT, and she is here now. And she's here with her family, sorry. And this is very important because her family, her children, Hugo, they are very important because they are the future. So for me, it's very special to see children here, no? Because it's the future. And also with Nat, with her husband that has the company Pumpkin Seed. So it's also important for SHIFT here, not only academic, but also people for the enterprise and for the spin-off companies and things like that. So the knowledge transfer is very important. So I finish here. I give the floor to Jennifer Dionne. Please give a warm welcome to Jennifer. Thank you.
All right, well, good morning, everyone. First of all, thank you so much, Jorge, for bringing us all together in this beautiful island. It's such a pleasure to be here. And as Jorge mentioned, I'm here with my family. They are having so much fun on this island, checking out the beaches. They're excited to go to the volcano. They're excited for the music and talks tonight. So thank you for having us here. And thank you also for this really wonderful cultural presentation on Blas Cabrera, really an inspirational person for physics in Spain. Today, I think Jorge said we still have the full 50 or so minutes, even though we got started a little bit late. And what I wanted to share with you is a bit of a story about the interaction and intersection between light and life, and how we can use that interaction for improved energy and biomedical applications. So first I wanted to start with a picture of my two kids when they were exploring the last volcano they were on. This is at Lassen Volcanic National Park in California. A beautiful place. If any of you ever come out to California, please let me know so I can host you at Stanford. But also be sure to check out some of our incredible national parks. And this is at a particular hike where they have sulfuric lake beds, of which I understand there are also several in El Teide National Park, and hopefully we'll see them tomorrow. And the reason I show this picture is not so you can just adore my beautiful, cute kids, Marcus and Hugo, but also because it's hypothesized that some of the building blocks of life actually came from these sulfuric lake beds, so these prebiotic lake beds. In the early Earth, basically had magnetite floors, right? The Earth has this magnetic crust. And it's hypothesized that amino acids, the building blocks of proteins, have a certain handedness because of the way ultraviolet photons from the sun interacted with this magnetic floor bed and gave rise to spin-polarized electrons that then got injected into the molecules into these sulfuric lake beds and gave rise to a certain handedness, namely a left-handedness or chirality, of all amino acids. And then, of course, we know that since amino acids are the building blocks of proteins, and proteins are the functional molecules of biology, that ultimately those proteins led to cells. So ultimately, those proteins led to the first cyanobacteria. These are photosynthetic bacteria that could take basically the carbon dioxide-rich, methane-rich, ammonia-rich environment, all those gases, and convert them into oxygen. And nowadays, photosynthetic bacteria, these cyanobacteria, are only about 1% of plant biomass, but they're responsible still for over 50% of Earth's photosynthetic activity. So here we have two examples of how light interacting with materials gave rise to life itself. And then, of course, in the early Earth, once we had these cyanobacteria and they started photosynthesizing, they were able to take this oxygen-devoid environment and turn it into an oxygen-rich environment. And this is something called the Great Oxidation Event, where if you look at the amount of oxygen in the atmosphere, you can see shortly after these cyanobacteria were formed, we had an abundance of oxygen that entered our atmosphere, and ultimately that's what gave rise to all of the plants and animals that we now see today. And nature, through these plants, basically learns how to make very useful chemicals for its own productive purposes. And these chemicals include, for example, ammonia. So ammonia basically comes from an enzyme that is in a bacteria that is symbiotic with plants. And the enzyme in the bacteria is called nitrogenase. And it fixes nitrogen to create ammonia by taking sunlight and then using the active center of the enzyme basically to create ammonia. And of course, ethylene, basically the precursor of polyethylene, one of the most common plastics, also found basically as or created by an enzyme that is in basically fruit-bearing plants and trees. And then phytoplankton, also some smaller organisms found in the ocean, are responsible for creation of methanol. So these are super useful chemicals. We now make them industrially, but nature learns how to make these at ambient conditions using just sunlight to drive the production of these chemicals. And then lastly, nature also learns how to efficiently communicate information. And since we just heard this amazing cultural presentation on physics, I'm including here a calculation that was published a few years ago by Lingham and colleagues in the journal Life, where they used principles of thermodynamics to determine a lower bound on the rate at which nature communicates information with itself. Basically, signaling molecules. How is nature communicating information? And they estimated that the transmission rate of the biosphere exceeds that of the technosphere by about 9 orders of magnitude. So to me, this is kind of amazing. Basically, it means that if you think about all of our fiber optic communications, telecommunications, computing, nature is still communicating more efficiently and 9 orders of magnitude more than we are in the technosphere. So what we're doing in our lab at Stanford is trying to figure out how to learn from nature, not only to make useful chemicals, but also basically to get inspired by the information transmission rate and hopefully improve better sustainable technologies and also better diagnostics and therapeutics. So what I want to share with you today is 2 vignettes from my lab at Stanford for how we're trying to better extract this data. And for those of you who haven't been to my lab at Stanford, I do hope you'll get to come visit soon. But generally, we focus on new ways of using light both to detect molecules and to direct their transformations. So part of my lab focuses on novel ways to use light to synthesize materials in a more sustainable way for chemical manufacturing. And that includes both small molecule synthesis as well as macromolecular synthesis. And then we also try to use light for molecular sensing and trying to get new data that is really challenging to get with other methods, or at least challenging to get in a high throughput and high data velocity way. So as I mentioned, I'll share with you 2 vignettes focusing first on small molecule synthesis and how we can use light coupled with industrially relevant catalysts to drive sustainable chemical manufacturing in a way that's inspired by nature. And then the second part of my talk, I'll share with you how we're using light combined with silicon photonics to extract new information about biology that has been prohibitively challenging with state-of-the-art methods. So foremost, I don't have to tell you that the world is consuming and emitting enormous amounts of greenhouse gases. If you look at kind of the sector breakdown for the largest contributors to greenhouse gases, there's of course the power, like the heat and electricity industry. But there's also industry, which here on this plot corresponds to the chemical manufacturing industry. And then as you go down, you can see mobility, buildings, agriculture, forestry. Those are also contributing substantial greenhouse gas emissions. And you can see the sources of those emissions are coming from, say, oil, natural gas, and coal. And I really admire Europe for its dedication to sustainability and alternative sources of energy. But I was pretty shocked looking at this plot at just how much the chemical manufacturing industry is producing in terms of emissions. And if we further break down that roughly 19.2% of global greenhouse gas emissions, you can see in the chemical manufacturing industry, roughly 8% comes from the iron and steel industry. Another 12% comes from different chemicals and plastics, including the refining process. And then even just to make fertilizer, we're consuming or emitting roughly 3% of all global greenhouse gas emissions. And roughly half of the nitrogen in our body actually comes from the Haber-Bosch process to create the fertilizers that we use to fuel the world. So all of these are really critical processes, and yet contributing considerably to greenhouse gas emissions. And the reason why is because, of course, we're using industrial processes to make these chemicals that I just shared with you nature was able to do in the early Earth at ambient conditions. So just using sunlight, room temperature, ambient pressure conditions to be able to create chemicals like ammonia, ethylene, and methanol. And yet in industrial processes, we need basically cities, that appear with like miles and miles of tubing to be able to operate at very high temperatures and very high pressures to be able to create the chemicals that the world needs basically to sustain its economy. So what we try to do is look to nature and gain inspiration from these natural proteins, these enzymes that are able to basically create these valuable chemicals using light at ambient conditions. And basically what happens in these enzymes is instead of being in thermal equilibrium, they operate out of equilibrium. So I love the speaker Monday morning who was talking about don't live in equilibrium. We want to be out of equilibrium because if you're in equilibrium, you're dead. But when you're out of equilibrium, you can generate all sorts of new products and basically new transition states that simply are not possible when you're operating in thermal equilibrium. And to illustrate how that works for a catalyst, if you're in thermal equilibrium, you have your industrial catalyst, which is generally going to be an inorganic metal nanoparticle. That is going to have some molecular reactants that are absorbed on the surface. And then you want to turn those into products. So there is going to be some reaction coordinate that brings you from your reactants to your products, usually overcoming an energy barrier through increased temperature or increased pressure. But once you're out of equilibrium, so once you turn the lights on and you're photo-exciting your system, now you have the ability to excite hot electrons or hot holes. You can excite excitons. You can excite plasmons, collective oscillations of the conduction electrons in the metal. And that can bring your reaction coordinate from the ground state to an excited state where you have totally new reaction intermediates that become possible and new reaction pathways. Here showing just a simple reaction pathway as you're going from the reactants to the products. So why is it that you get these new reaction pathways? Well, part of it, like I said, is because light is coming in and exciting these hot carriers and these plasmons. And for those of you who aren't familiar with plasmons, they are these conduction electron collective oscillations that are occurring in these small inorganic metal nanoparticles. And compared to the conditions that you would normally be able to control at the bulk level, like pressure or temperature or electrical potentials or optical potentials even without plasmons, those generally are occurring on the bulk scale, whereas plasmons can be controlled on the nanoscale. So we have the ability to control the distribution of light and temperature and electrons as carriers to drive new reactions on length scales that are comparable, if not smaller, than the catalytic nanoparticle itself. And that gives us the ability to almost have a molecular scalpel with which to control molecular reactions that are occurring on the catalyst surface. And just to illustrate that in a little bit more detail, there's been a long history to this field, but with plasmons, so optical excitations of metallic nanoparticles, you can control the scattering or the color of those nanoparticles. So you can take, say, a silver or a gold or a palladium nanoparticle and tune the color simply by tuning the shape of the nanoparticle. So here what we're looking at is basically the color of light that gets scattered for a certain metallic nanoparticle, just changing the shape of it. And then if you go in with an electron microscope, you can actually probe how light is being controlled on a length scale that's comparable to the metallic nanoparticles. So here is actually an industrial catalyst that's used for creating ethylene. It's silver plus palladium. And you can see that the silver and palladium are alloyed together almost homogeneously, so we can figure out the elemental composition. But then if you look at how light gets concentrated at different wavelengths or at different energies, in this case 3 electron volts, 5 electron volts, and 7 electron volts, you can see that visible light gets concentrated either to the tips, if you're at lower energies like 3 electron volts, or to the edges if you're at 5 electron volts, or kind of all over the surface if you're at even higher energy, 7 electron volts. So these are experimental maps of how light is getting concentrated to deeply sub-wavelength scales on these nanoparticles. And then if you start seeing how these optically excited nanoparticles impact catalysis, like I mentioned, this is a pretty long and vibrant field, been around for at least 20 years at this point. But it's kind of amazing that you can start to see increased reaction rates just by illuminating your reactor bed or your catalyst bed while the reaction is occurring. And then what I think is most exciting in this field is not so much the increased reaction rates, but the ability to tune the yield and the selectivity of your reaction. So this is some work done by Dane Swearer, who was a PhD student with Naomi Hollis at Rice University. He then came to my lab as a postdoctoral fellow. But here he was looking at acetylene hydrogenation to create ethylene, basically a precursor to polyethylene. And if you look at traditional thermocatalytic processes, using again those silver and palladium nanoparticles that I just showed you, basically the yield of the ethylene you get basically goes up, showing the increase in temperature in black here, while the selectivity in red generally goes down. So as you increase temperature, you can get more product, so your yield goes up, but you actually get less of the product you want. The selectivity for creating ethylene, as opposed to other products that sometimes can be poisons to the downstream catalyst, basically there's a trade-off there between yield and selectivity. But interestingly, he found that when he illuminated his reactor bed, that trade-off went away. Both the yield and the selectivity kept going up as he turned up the laser power. So this is showing that it's not just by illuminating that you're controlling the local temperature. There's actually something more going on. And in terms of what more is going on, the hypothesized mechanisms span the fastest of time scales and the smallest of length scales all the way up to the reactor scale. So of course I mentioned you have these hot carriers. These are basically non-thermal distributions of electrons and holes, basically that are being excited to energy levels that are distinct from the Fermi-Dirac energy distribution in the nanoparticle. And then those hot carriers can be directly injected into HOMO and LUMO levels of whatever your molecular absorbates are during the reaction to drive particular reactions. Then, of course, you have the near-field enhancement, the increased local density of optical states that can give rise to kind of modified electronic energy levels, modified thermal energy levels. And that all has to get mapped onto site selectivity. So intrinsically, what are the sites that are most active for catalysis on the particular catalytic particle you have? And then we're not just looking at these catalysts in isolation. You are putting them into reactors with a certain mass loading. So you have to think about how the catalytic nanoparticles are interacting with each other to give rise to collective heating effects, and how that heating plus whatever optical illumination you have might be propagating throughout the entire reactor bed, which can range anywhere from a benchtop reactor to something like that city, skyscraper that I showed you earlier in the presentation, where you have like a 10-story building that is your reactor. So in our lab, what we try to do is link these length scales, looking at the smallest of length scale, so the atomic scale, to think about things like site selectivity and how we have local optical excitation and how that gets mapped onto function. And then we try to also map that onto what happens at the reactor level. So in the next couple of minutes, I'll share with you our development of environmental electron microscopy. Probably many of you have heard of cryo-electron microscopy. Environmental microscopy is a pretty amazing technique where instead of working under high vacuum, basically the system is differentially pumped such that you can have almost atmospheric pressures, if not higher, at the sample chamber in your electron microscope. So we can introduce different gases. We can also modify the temperature. And then what we've also been able to do is introduce light into the electron microscope so we can see how going out of equilibrium changes basically the catalytic or chemical transformations compared to being in equilibrium. And then, like I said, we try to link that to reactor scale properties. And I don't know if anyone has done catalytic reactions here. If you do, we have an open source software that allows us to automate a lot of our reactions so we can vary the illumination wavelength, the illumination power, the temperatures, the pressures, and really explore this fairly complicated and complex phase space so that way we can optimize catalysts in a rational way by using our TEM-informed insights and then also looking at what happens at the reactor scale. And the reactions that my lab are most interested in are actually related to the ones I showed you that nature was able to do so efficiently. So ammonia synthesis to create fertilizers via the Haber-Bosch process in industry, but can we learn from those nitrogenases to make it at ambient pressures? Also CO2 reduction, how can we reduce carbon dioxide and turn it into useful chemicals? And then also methane coupling. Methane is actually one of the principal components of natural gas. Can we activate methane and convert it into high-value chemicals? So you can see basically from these reactions that they all involve hydrogen of one form or another. So when we were first thinking about how to link basically the atomic scale to the reactor scale, we looked at these hydrogen-based reactions. And that's because hydrogen intercalation at the surface and subsurface of these nanoparticles is a really important deciding intermediate step in a lot of these critical reactions. And just to show a few examples, basically if you have hydrogen, H2, basically it catalytically splits at the surface of your metal nanoparticle. And depending upon how much hydrogen gets into the surface and into the subsurface of your catalyst, you can strongly influence the outcome of your reaction. So here are just a few examples showing, for example, that the catalysts that have been used for, say, CO2 reduction have lower activity once there's too much hydrogen that's intercalated into your nanoparticle. And you can also modify the selectivity. So I showed you earlier how with light we can control the selectivity and the activity. Here this is showing basically as you're controlling the pressure of hydrogen in your environment, you actually get an additional knob over the selectivity. So what we did is put some of these catalysts into our environmental transmission electron microscope and then just wanted to know how do they behave when there's only hydrogen. So there are no other reactants in this particular case. And to probe what happens with hydrogen, we use both electron diffraction as well as spectroscopy. So electron energy loss spectroscopy, basically you're shooting your electrons, 300 kiloelectron-volt electrons, through your sample, and then we have an electron spectrometer that can measure down to about 10 millielectron volts difference, which is kind of amazing if you think about a 300 kiloelectron volt electron, and then we're looking just at tiny differences, tiny energy losses that are down to about 10 millielectron volts. But both of these give us an indication as to a change in the structural property. So hydrogen, when it intercalates into many of these materials, is kind of a misfitting sphere in those interstitial sites, those voids. So it tends to expand the lattice. So from diffraction, we can look at structural changes. From the electron spectroscopy, we can basically look at electronic changes. So I won't go through the details of how we do all of the experiments, but here was one of the early experiments now from, I guess, about almost 10 years ago, where we were actually visualizing the kinetics of hydrogen intercalating into a pure palladium nanocube. And you'll see as the talk goes on how our resolution has improved. But if I play the video one more time, hopefully it'll let me. Maybe I'll go back one slide. Here we are. You can see that the corners of the cube start turning white. That's actually where hydrogen is starting to intercalate into the cube. Eventually, the hydrogen starts to linearize across the cube and then propagates across the entire cube until it's fully hydrogenated. And if we look at some still snapshots of that particular nanocube, we can combine both this direct imaging, which basically comes from dark field imaging and diffraction, with electron spectroscopy to confirm that the new hydrogenated phase, which I've labeled here as beta, basically starts at the cube corners. So it makes sense. Those low coordination number sites, those corners, are the most active sites for hydrogen intercalation. And then hydrogen intercalates into your nanoparticle, stabilizing along this 100 type interface until eventually the whole cube is hydrogenated. And what we found in the past decade is that regardless of your nanoparticle shape or size or crystallinity, basically the hydrogen is always going to split and intercalate at those low coordination number sites. So that was a cube that I showed you with 100 terminated facets or faces. Here is an octahedra with 111 terminated facets. And again, you can see the new phase intercalating basically from the octahedral corners. Similarly, for these nanorods, we also have hydrogen basically splitting at the tips and then going into the rod. So when we are in equilibrium, we don't have any lights on, but we're just controlling things like the temperature and the pressure. Regardless of the conditions, we always have the hydrogen splitting and intercalating into these nanoparticles at the cube corners. So a few years ago, we wanted to know, okay, what happens when we start to turn the lights on? Might we have the ability to control where the hydrogen is intercalating? And can plasmons, those collective oscillations of electrons, actually activate a non-active site? So perhaps we can move away from some of those low coordination number sites. So we set up a kind of a toy experiment where we had a gold nanobar and then a palladium nanorod on top. And this was a nice experiment because even though we have roughly nanometer scale imaging spatial resolution, we could still resolve the difference between the tips and the middle of the rod with very high accuracy to determine where the hydrogen was nucleating. And then propagating in the particle. So you can see here that we have an electromagnetic hotspot, basically a region of very strong confinement between the gold bar and the palladium nanorod. And we wanted to know if that region of strong electromagnetic confinement would basically change where the hydrogen was intercalating and deintercalating. So this is something that is called an antenna reactor system. So we have light as the strong, or gold basically as a strong light absorber. And then we have palladium as the catalytically active nanoparticle. You can see that this particular system has basically the predicted near field enhancement over the junction between the gold bar and the palladium nanorod. And basically it absorbs light most strongly at visible frequencies in the red, so right around 600 or so nanometers. And hopefully this video plays. I think in the back they might play it. Basically what we're doing here is doing dark field imaging and diffraction. I don't know if you're able to click play on this video, but it seems like it's not working yet. Oh, nope, the video did not play. Sorry, maybe you can go back once. Here we go. Okay, so we're doing dark field imaging and diffraction, looking at one of the crystallites in this palladium rod. Here's the gold bar. White corresponds to where the system is hydrogenated. So we turn the light on, and now you can see the hydrogen is basically leaving from the center of the rod. And then as the video plays, we'll turn the lights off, and you'll see that hydrogen is basically able to enter the system again, but it does so from the tips of the rod. So you can do this over and over again, and basically you can nucleate where hydrogen is entering the rod and where hydrogen is leaving the rod simply by using the light to trigger the reaction. So with the light on, we basically trigger the reaction at that electromagnetic hotspot. With the light off, basically we only have hydrogen entering from the tips. And here we did this as a function of the illumination wavelengths. So you can see only when we're illuminating right around 600 nanometers do we actually get this effect. So you have to be at that plasmon resonance of the nanoparticles. Otherwise, you basically get no nucleation regardless of what wavelength or what power you're illuminating with. So integrated over a large number of experiments, of which this is one example, what we've learned is that basically you have hydrogen that is entering the surface and subsurface of your catalytically active nanoparticle, and light almost becomes a new reagent with which to control the absorption and desorption of hydrogen at the surface and subsurface. So basically, light gives us the ability to control hydrogen on demand. So how can we use these sorts of insights to enable production of valuable chemicals? That's where I'll turn to just this short story, which is using one of those antenna reactor systems, but here as a bimetallic nanoparticle, and this is more similar to what you would find in industry, where in this case we have a catalyst that's comprised of ruthenium, so that's the catalytically active material for creating ammonia. But what we did is alloy together ruthenium with gold. So gold is our optical absorbing material here. And the idea was, can we use the plasmon resonances of gold in this catalytic system to basically drive ammonia synthesis at room temperature and ambient pressures instead of requiring 20 megapascals of pressure and 500 degrees C. And the industrial Haber-Bosch process usually relies on ruthenium and sometimes on iron. So we basically swapped out the iron for this gold. So here you can see basically the extinction cross-sections where the particle is absorbing light. Again, it's at visible frequencies, in this case between 500 and 600 nanometers. And what we did is basically change the amount of concentration that was in our alloyed nanoparticle with a gold ruthenium composition that had about 20% ruthenium and about 10% ruthenium. And if you're doing just thermocatalysis, so the reaction in the dark, here you can see as you increase the amount of ruthenium, your reaction rate for creating ammonia from nitrogen and hydrogen actually goes up, and that's what you'd expect because you have more active sites to basically drive the reaction. Similarly, here, under illumination, as you increase the number of active sites, basically that reaction rate also goes up quite substantially. But now, if we go up to 30% ruthenium, you'll notice that the reaction rate actually goes down for photocatalysis, up for thermocatalysis. So this means there's kind of a Goldilocks effect where when you're doing photocatalysis, you want to have a strong enough optical absorption. And in this case, the ruthenium kind of quenches the optical absorption. But in either way, when we have basically this optimized system, where we have 20% ruthenium, even just with one sun illumination, room temperature, and room pressure, we have almost twice the reaction rate as you can get with higher temperatures and higher pressures. And what we've done is also combine these sort of reactor scale experiments with in situ FTIR spectroscopy to look at the intermediates. And I'll just very briefly go through these sorts of intermediates where, for example, we're looking at the NHX adsorbate densities and also the NH3 adsorbate density. So here what we're doing is basically looking at different intermediates at different time scales during the reaction. And what you can see is for this particular intermediate, where X is either equal to 1 or 2, whether you're in the dark or with illumination, basically that intermediate isn't changing. But you can see here, when you're heating versus just using light illumination, this NH3 adsorbate density changes quite dramatically. So what we're able to do with light is basically change the reaction pathway. And that's illustrated here by some density functional calculations where we do both the ground state calculation shown in bold and the excited state calculation shown in the dashed line, and basically look at different pathways and how those are modified. So here we're looking at the N2 dissociation mechanism and then the hydrogen-associated dissociation mechanism of that N triple bond N. And to break that N triple bond N, which is often thought to be the rate-limiting step in ammonia synthesis, you can see whether we're in the ground state or whether we're in the excited state. First of all, we've changed the intermediate energy levels, but the energetic barrier to go from N2 plus H2 to this N dissociated bond is still quite substantial. It goes from 6.2 electron volts to 4.3 electron volts when you're in the excited state. So with visible frequency light, we're still not going to be able to overcome that activation barrier. But now if we look at this hydrogen-assisted splitting of nitrogen, in the excited state, we bring that energetic barrier from what used to be about 2 electron volts down to 1 electron volts. So we're actually changing the reaction pathway, and by illuminating at visible frequencies where we have over 1 electron volts, basically we can overcome the barrier to go from this excited state to this intermediate. So basically we're using light to drive the hydrogen-associated dissociation of nitrogen. So basically what this means is that it is this hydrogen-assisted splitting of nitrogen in the excited state that's the key mechanism for how we're getting this ammonia synthesis at room temperature and room pressure. And it's also interesting that people have hypothesized that this is the same way that the nitrogenase enzyme works. So I think it's neat how now insights from physics and inorganic chemistry might actually inform how nature is working as an enzyme. Okay, so in the final few minutes before I switch to some bio applications, I wanted to show you what might be happening on the surface of these catalytically active nanoparticles. And at least what's hypothesized is that when you have a nanoparticle in a certain size regime, you basically have these step terraces that correspond to basically B5 sites. So a B5 site would be 3 ruthenium atoms in one layer and 2 in the layer above. So these are the hypothesized sites by which you basically have the appropriate spacing between, in this case, nitrogen and the hydrogen. Here, the hydrogen shown in white, which basically has split into a hydrogen atom. These are basically plots from Jens Norskov, who did some really beautiful DFT to try to understand what the active sites might be and what are the energetic barriers for forming nitrogen on each of these active sites. But to date, it remains a theoretical construct, and it's also not known how these active sites might change when you illuminate with light. So you can illuminate with light and potentially even change basically the interatomic separation between your catalytically active sites on the surface. So what we've started to do now is use our environmental transmission electron microscope to do atomic electron tomography. So what we can do is take images of our catalytically active nanoparticles at different tilt angles. Each of these has atomic scale resolution, and then at different tilt angles, we can figure out basically what is the three-dimensional construction of the catalytically active nanoparticle. So here I'm showing you the bond length between each of the atoms in this particular ammonia catalyst. And then we can also look at the coordination number. So basically how many other atoms is a particular ruthenium atom bonded to. And if you look at the experimental tilt series and then do the atomic reconstruction of what the 3D structure of your catalyst looks like, and then take that atomic reconstruction and back project it to each angle, you can see that that simulation or that back projection actually has amazingly good agreement with what we experimentally observed. So we know that this atomic 3D construction corresponds quite well to what we're experimentally seeing. And we can do this over different size nanoparticles and different composition nanoparticles. And what we start to see is we can look at the ruthenium-ruthenium bond length, the coordination number, and then from there basically extract what are the B5 active sites. And what we're seeing is that when we have catalysts that are somewhere between basically the 2 and 3.5 nanometer regime, basically about 20% of the catalyst surface corresponds to those B5 active sites. So now what we can go and do is drill down into those active sites and basically see how they're being changed when we have hydrogen and when we have nitrogen in the environment. So to summarize kind of where we are so far, we've built up a pretty amazing capability, which I encourage you all to come and hopefully collaborate with us on, which is environmental electron microscopy that has optical excitation capabilities, where we're able to look not only at hydrogenation now, but at more complex reactions like ammonia synthesis in the transmission electron microscope. And here are just some videos of the sorts of data we've been able to get. I was able to show this incredible individual, Rupert Stryer, a tour of our TEM facilities before he passed away. And the title of my talk is actually inspired by this really incredible article he wrote back in 2012 called Exploring Light and Life. So Rupert Stryer, I think, was a pioneer of really understanding the interaction between light and life. So I encourage you to read this article. And after he visited my lab, he sent me this very nice email, of which he said, since I just received a fellowship, I hope you'll be able to use that funding to pursue your most challenging and riskiest ideas, those that can most profoundly influence the future course of science, engineering, and medicine. And since many of you are students, I hope you can use this time as a PhD student basically to explore your most challenging and riskiest projects and those that can most profoundly influence the future of humanity, because I think we're at such an interesting time now. The world is filled with opportunities, but I also think the timing is critical to address some of these big challenges in sustainability and also in health. So with his advice, I wanted to transition basically to an area where I'm quite excited about how the work that we're doing in the lab can actually translate into commercial applications. And in this case, instead of focusing on sustainable chemical manufacturing, we're focusing on basically health applications and the amount of data that it takes to really drive new insights for drug discovery and for better medicines. And if you think about how biology communicates information, it's not at the DNA level or at the RNA level. So the genome and the transcriptome, in a way, are kind of like the blueprints, but the working molecules of biology are your proteins. And then, of course, you also have metabolites and lipids. And yet, most of the data that we have available to inform health and transitions from health to disease basically is at the genomic and transcriptomic level. So what my lab has tried to do in the last handful of years is get more information at the proteomic and peptidomic and lipidomic level to basically help inform new therapeutics. And part of the reason why this is so challenging is because there's not a one-to-one mapping between the genome and the proteome. So in our bodies, we have about 20,000 protein-coding genes, and yet there are at least millions of possible proteoforms, estimated to be about 10 to the 27 possible proteins. So it's a one-to-many mapping. So just by knowing the genome, you cannot predict what proteins are present and how those proteins might be changing in health and in disease. So what we do is, again, use this kind of light-inspired toolbox to try to get information at the proteomic, lipidomic, and metabolomic level, and in particular using Raman spectroscopy. So I shared with you during the catalytic part of our talk how we're doing in situ FTIR spectroscopy to try to learn about the intermediates and how those are changing with illumination and with temperature. What we do in the biology case is basically, again, use this vibrational spectroscopy to get label-free information about whatever biomolecules are in a particular sample. So the key gist is we come in with a monochromatic light source, usually a diode laser. And then we have our biological sample that's introduced to basically a silicon photonic chip that has millions of sensors on it. And those sensors are designed to strongly enhance Raman scattering, at least by many orders of magnitude, usually about 8 orders of magnitude. And then we're looking at this vibrational scattering signature, so this Raman spectra, which is a label-free spectra, but incredibly information-rich. So we're getting information about all of the molecular vibrations that are in a sample. And then what we're able to do is not only boost Raman spectroscopy to, in some cases, the single molecule level, and certainly the single cell level, but then also gain interpretability of that Raman spectra by looking at different vibrational peaks and collections of peaks to provide interpretability as to what sort of molecular vibrations they correspond to. So I'll spend a few minutes just discussing the particular sort of chips that we're using to enhance Raman spectroscopy. I would say all of them tend to be close cousins of one of the first high-quality factor metasurfaces that we made in our lab. It's basically silicon nanoresonators, in this case silicon bars, that have slightly different widths in this cavity region, which here I've labeled as L1. And then we have basically a tapered photonic crystal mirror that makes the structure appear as if it's infinitely long, even though it's just going to be a few microns long, and then kind of this padding region. And what we do is take these silicon bars, we use CMOS-compatible fabrication processes to pattern these on a substrate. We illuminate anywhere from the visible to the near-infrared. And then what we do is in the far field on a CCD camera, basically detect the Raman scattering signal. So these structures are designed to have a high quality factor, meaning that light goes from the bottom, gets trapped into this almost waveguide type propagating mode for a long period of time. So quality factor is a measure of how long light gets trapped in the structure. But then the structure is also designed to confine light to extremely wavelength and sub-wavelength scale. So we have a high quality factor and a small mode volume. And based upon the size of these structures, we can actually pattern millions of these per square centimeter in the clean room. And why do you want a high quality factor and a small mode volume? Well, the Purcell factor, which basically governs the efficiency of dipole emission or Raman scattering, is directly proportional to your electric field enhancement squared, which is also proportional to the wavelength cubed times the quality factor divided by the mode volume. So generally, it's been thought, well, you can either boost Q or decrease V. In our case, we actually try to do both. So we try to get high quality factors and small mode volumes to get the highest possible Purcell factors. So those width perturbations or sometimes height perturbations that I showed you in the schematic of that dog bone type resonator structure give rise to extremely high quality factors. The quality factors can be in the millions based on how you're tuning the geometry. And here's a scanning electron micrograph of just one example system that we've made in our lab. And then I also mentioned, in addition to the high quality factor, we can shrink down the mode volume. So here's an example of one of those structures where we've basically kind of patterned a slit into the middle of it. And you can see how this is acting almost like a lens to focus light to the nanoscale. In this case, apologies for not including a scale bar on here, but here we have extreme mode confinement. So each of these bars is just about 200 nanometers across. So basically the mode volume is about 10 or so nanometers cubed, so extremely intense electromagnetic fields in a very small mode volume, but in this case using silicon. So silicon is not absorbing in the near infrared, which means that you're actually not heating up your system. So if you functionalize your surface for the biomolecule to land there, you're actually going to be able to do dynamic and longer term interrogations because you're not heating up your biomolecule. Basically, the strong electric field is coming from that high quality factor and how light is just propagating many optical cycles in that small region. And then here's a video of the sort of experimental data we get. So here we have about 300 sensors. We're just illuminating them in the near infrared. And you can see each of them is basically lighting up at different frequencies. So basically, we're scanning the frequency of detection. You can think, OK, as we have different molecules landing on each of our sensors, we're collecting a distinct Raman spectra from each of these resonators. So it's kind of neat that there's no fluorescence here, right? We're simply looking at the optical scattering of the resonators and how those resonators are interacting with whatever molecules or whatever materials are in the near field. And from the particular pattern of the far field scattering we see, we can relate that back to whatever molecules have landed on our sensors. So in Stanford's clean room, we were able to pattern about 3 million sensors per square centimeter. Depending upon how you functionalize each of these resonators, basically you can use bioprinting to locally functionalize each of them. So for those of you who do biology, basically the number of sensors we have on this chip corresponds to at least 31,096 full plates. So in a way, it's kind of doing lab on a chip, where if you were to try to do this amount of biology, not only would it take you a lot of time for some of the applications I'm going to share with you and an enormous amount of capital, but you would also need what we can do on a 1 centimeter square area, roughly the size of a basketball court. So I don't know if you can see kind of the purple outline of the resonators here on the chip that my student Varun made, but each of these has high quality factors and small mode volumes. In this case, experimentally, the quality factor was a little lower than what we can do now, but still it was over 1,000 here. Okay, so what we do is basically functionalize each of our resonators so that way biomolecules can be attached. In this case, we were attaching just a sugar molecule in the interest of doing glycosequencing. And here I just want to share with you that when we have the right polarization of light, you can see the Raman intensity of this particular molecule is quite strong to when we have the wrong polarization of light. And then similarly, when we are on resonance, so at that high quality factor resonance, that basically boosts the quality factor, but the mode volume at the pump wavelength, you can also see that the Raman scattering goes up quite substantially, whereas if you're just off that high Q resonance, you basically don't get any Raman scattering signal. And then what we can do is collect Raman spectra from many of our resonators. Here's the experimental data. What we wanted to know is, can we predict theoretically what the Raman spectra is going to be? And in the interest of time, I won't be able to discuss the workflow that we've developed for doing first principles calculations that look at both DFT and molecular dynamic calculations to figure out what the Raman spectra is going to be of very complex molecules, or at least macromolecules. But here for this particular sugar, when we collect the Raman spectra, basically what we do is figure out what are the different conformations, and then how do those conformations give rise to different Raman scattering signals. So for this particular sugar molecule, you can see that there's actually 2 different conformations, here shown as those 2 different clusters. And when we're doing these dynamic calculations, we can see that one of the clusters, basically that conformation is present about 40% of the time, 39.7% of the time. The other conformation is present about 60% of the time. Each of those conformations has distinct Raman spectra. And when we take each of those Raman spectra and then do a weighted average for 39% of conformation 1 plus 60% of conformation 2, basically we get a theoretical prediction that matches almost perfectly with our experiment. So since experimentally we're integrating for roughly tens of milliseconds, we're getting this kind of ensemble average of different conformations of the Raman spectra of this molecule. Okay, so just in the final few minutes, I want to share with you now how we're applying this to what I think are pretty compelling problems in health and in medicine. And some of these, in fact, this one relates to a spin-out company from my lab that some of you heard Nyat and Jack speak about on Monday's panel. Okay, so if you think about how all of our cells are working right now, we have proteins as the working molecules, the functional molecules of biology. Those proteins are in our cells. Most of our cells are nucleated. And basically, all nucleated cells have a process for chewing up those proteins and then spitting them out as fragments that are on your cell surface. And those fragments of proteins are called peptides. And they serve as beacons for your immune system to tell if a cell is healthy or diseased. So your immune system, including your T cells, will kind of go around trying to feel out these protein fragments and determine if the protein fragment is a healthy protein fragment or if it is a diseased protein fragment. And usually the diseased protein fragment corresponds to just a single point mutation, so a single amino acid that is mutated in that protein fragment. And I like this review from Nature Cancer a few years ago that says each TCR, which is a T cell receptor, so each receptor of your T cells as your immune system can bind to a particular, albeit unknown, set of antigens. Here, antigen means protein fragment or peptide. So our T cells are amazing. They can bind to pretty much any antigen or any peptide and figure out when you're healthy, if it is diseased or if it is a healthy peptide. But these peptides are mostly unknown. It is extremely challenging to figure out what these protein fragments are on cell surfaces. And the reason why it's so important basically to figure out what these peptide fragments are is because as you get older and your immune system is less good at recognizing those disease peptides, there are biotech companies that are now trying to figure out these peptide sequences so that way they can give you a peptide therapy that basically helps you to prevent metastatic disease. So if you have cancer, they can remove the cancer. Usually you will not die of the first cancer you have. It's the metastatic disease that gets you. So the idea is, can you figure out on an individual patient tumor what those peptide fragments are, and then give the individual a therapy that teaches your immune system to fight off that particular mutation. Okay, so what we're doing is basically this Raman spectroscopy aided by these silicon CMOS compatible resonators that boost the electromagnetic field, so that way we get Raman enhancements by at least 8 orders of magnitude. And the idea is that you can take these peptides, remove them from a sample, like a patient tumor sample, and even though the number of peptide sequences is extraordinarily diverse, they all have a C-terminus and they all have an N-terminus. So that C-terminus can be bonded covalently to the surface of our chips just using click chemistry. So we orient the peptide, and then what we do is we collect the Raman signal dynamically as we're cleaving off the N-terminal amino acid. So this is the company Pumpkin Seed, spun out from my lab. And basically, as we are looking at changes in the Raman signal before cleavage, during cleavage, basically when a particular reagent comes in and does just one amino acid residue cleaving at a time, and after, we're able to uniquely determine which amino acid has been cleaved off, at least for these immune-related peptides. So here is an example of the chip in a flow cell, where in the flow cell we can basically control the temperature and the pH and introduce different reagents for this cleavage. You can see here how the Raman spectra is changing as a function of time. And then using this process, we've been able to differentiate both wild type and mutated peptides. So here are example spectra. Each spectra is a dot projected onto this two-dimensional plane. So we've collected the signal from each of our sensors for a wild-type peptide, basically in the dark blue, and a mutated peptide in the light blue. Your amino acids basically have all of the letters of your alphabet. So here you can see when there's an alphabet swap that corresponds to a single amino acid change. It's only that single amino acid that gives rise to basically the difference between health and disease. This is something that was very challenging for our collaborators to figure out. using state-of-the-art mass spectrometry methods. And we can also do so on individual patient samples instead of needing to pool patient samples together. So from this sort of data, we're able to build machine learning models that allow us to have over 96% accuracy on a per residue basis. And we can look not only at differences between wild type and mutated peptides, but also at different post-translational modifications. And I'll just mention that we now are making these wafers with IMEC, a foundry here in Europe, and then also collaborating with Jenny Lill at Genentech to help get them the data they need to develop better antigen-specific therapeutics, whether in oncology or in autoimmunity. So that way you can specifically target the particular cancer or the particular autoimmune disease that a patient has. So just the final minute, I'll say we've also moved from peptides to cells. This is a collaboration with my colleague, Dr. Amanda Karane, who wanted to know, could we use our metasurfaces to look at her melanoma patient samples and basically better predict immunotherapies? So with traditional immunotherapies, which have pretty much revolutionized the landscape of cancer care, there's actually no predictive biomarker of outcome. So for certain patients, immunotherapy works incredibly well. Whereas for other patients, roughly 30 to 50% of them experience severe adverse events. And she wanted to know, a priori, could she actually predict what the outcome would be? So we went from looking at just these peptides to actually looking at whole cells. And I'll kind of skip over some of the details of the work here. But in the tumor immune microenvironment, you have your cancer cells, you also have your immune cells. And what we were able to do is train machine learning models to identify both cancer therapy-resistant kind of phenotypes and how that looks in the Raman spectra, as well as immune cell phenotypes. And here are just two example immune cells, including from our innate immune system and our adaptive immune system. And hopefully some of you were able to hear Ariel's talk yesterday, where she talked a bit about looking at these phenotypic states. So this is the final science slide, where basically what we did is compare traditional workflows that have transcriptomic data to our Raman-based workflow, which is much faster and much higher throughput and label-free, and basically show that when we looked at historic patient samples and trained models to say, well, historically we know this patient is going to be susceptible to Nivo or Cabo as like different drugs, then we could look at a cohort of kind of 33 future patient samples that our model had never seen before. And in 30 out of 33 patient drug outcomes actually accurately predict how a patient was going to respond to different targeted therapies as well as different immunotherapies. So I think Raman is exciting because it's incredibly information rich. And just going back one slide, I'd say not only is it information rich, but it's also not a black box anymore. So with advances in machine learning models, you can actually drill down into the Raman spectra. So looking at each wave number and figuring out wave number importances, so that way you can start to correlate them to different molecular biomarkers that are in the cell. So I'll wrap up now. First of all, if you want more to read, we have a Physics Today article that came out last year that talks about nanophotonics for sustainability, including for energy and for medicine. So it's, I know today was a bit of a potpourri of 2 different topics. This one has 5 different topics in it, but hopefully you'll enjoy it. And then if you were especially interested in advances in electron microscopy, I was able to write this MRS Bulletin review on quantitative gas phase transmission electron microscopy, and not only what we're able to do now, but also where the future is headed. So I will sum up and say, hopefully if you take anything away, it's that the ability to use light and to control light at the nanoscale can give rise to really incredible insights into both sustainable chemical manufacturing and new data into biochemistry. We talked about optical excitation with an environmental TEM and how we can use insights from the TEM to inform plasma and photocatalysis. And then in the second part of my presentation, I shift from metals to dielectrics and shared with you how we can use light combined with CMOS-compatible manufacturing for high-velocity kind of proteomic and phenotypic data. And with that, I would like to thank all of the students and postdocs who made this work possible. I wouldn't be able to do it without them. Also, a big thanks to our funders, and thanks to Jorge for this incredible opportunity.
So we have time for just a couple of questions, please. Questions?
Yeah, we'll start with you.
Here.
Thank you for the very interesting talk. For the Raman experiments, I was wondering whether it could be useful to use photonic chips, say in silicon nitride, for example, to operate at shorter wavelengths.
Oh, amazing question. Yeah, I didn't put that in my presentation, but that is something that my lab has been doing. Silicon nitride doesn't have, on its own, quite the high refractive index that you need. Silicon has a refractive index of about 3 compared to 2.02 for silicon nitride. But what you can do is dope silicon nitride with a little bit extra silicon, so silicon-rich nitride, and that works extremely well to get you the high indices but still have low losses if you want to shift to visible wavelengths. So yeah, silicon-rich nitride is a really cool material. I'd also say another one that we're working now with IMEC on is carbon-rich amorphous silicon. Actually also has low losses and works quite well at visible frequencies.
Thank you.
Another question? Oh, Nazario?
Yeah, wonderful talk. Thank you for the presentation. Could you figure out that because you can also prepare or to go to chiral gold nanoparticles, for instance. So could you figure out the idea of using like light absorbers and mixed with circularly polarized light and so on?
Yeah, I think that's a cool idea. Everything here was with linearly polarized light, but well, I think probably you've even done some of this work and others in the audience, where depending upon how you structure your nanoparticle, or your nanostructure, you can tune particular hotspots based upon the polarization of light. So using circularly polarized light plus structures that either break in plane two-dimensional symmetry or 3D symmetry, I think can give you very unique control over chiral synthesis. So we've started to think about that a little bit, but I think it's a really exciting direction that even goes back to the kind of amino acid synthesis that the Earth invoked. Like, can you nanostructure kind of a chiral catalyst where you use the light plus the structure of the catalyst itself to get locally spin polarized electrons? So I think that's a pretty exciting area for future work.
Absolutely. Thank you.
Only one last question. First question, please. Last one. I'm sure we will have more time in networking to discuss.
Thanks, Jen, for the amazing talk. For the active site 3D reconstruction, it's really beautiful, but I guess that under dynamic conditions, under reacting conditions, it will be different and also probably very dynamic. So do you know what kind of time resolution you could have to be able to map it out under actual reaction conditions?
Yeah, oh man, amazing question. So I didn't put it in this presentation, but we've actually started doing that. So we have basically a protochips holder in our TEM where we can introduce nitrogen and hydrogen. And we've looked first not at quite the atomic scale, but even just with different temperatures. If you go to a high enough temperature, you actually see the migration of the ruthenium atoms out of the gold and eventually kind of forming a void. To answer your question, roughly we have about like millisecond time resolution right now with dynamic TEM. You could get much faster, and I think Vibka is doing some of that already. But I think it's super cool that we're starting to get near atomic scale resolution in these dynamic environments by using some of these holders. And then what you can think about doing is starting to look at, at the atomic scale, how those active sites are actually changing under different gas conditions or temperatures or illumination conditions. So right now we've done basically that measurement, not in the dynamic environment or not dynamically, more just statically. But I think kind of next step is like, how are those changing?
Okay, thank you very much. Thanks to the speaker again. Thank you, Jan. Thank you.